Hello, this is Eva Amson welcoming you to this Bite-Size Bio webinar, which today is sponsored by Molecular Machines and Industries. MMI is the leading provider of unique technologies for microdissection, micromanipulation and imaging, with a wide range of clinical and research applications. These include CellCut, which uses laser capture microdissection to isolate cells and tissue, as well as tools for microscopy-integrated whole slide imaging, capillary-based selective isolation of single cells, and optical tweezers to quantify biological forces. Today's presentation is titled Plant versus Virus: Comparative Transcriptomic Analysis of Single Turnip Mosaic Virus-Infected Microdissected Plant Cells. The webinar is being presented by Dr. Björn Krenz. He leads the independent research group Virus Interact at the Leibniz Institute, DSMZ, in Braunschweig, Germany. Prior to his current position, Björn was a group leader at Friedrich Alexander University in Erlangen, and before that he held postdoctoral positions at the University of Stuttgart and at Cornell University in the USA. Björn's current research at the DSMZ, or the German Collection of Microorganisms and Cell Cultures, aims to analyze and characterize the molecular mechanisms and interactions between plant cells and viruses. As always, we will have a question and answer session after the presentation. So please type any questions you have into the questions box, which will appear on the right-hand side of your screen, and I'll put them to Björn at the end. The recording of this webinar will be available at bitesizebio.com. So now over to you, Björn, for the presentation.
Hello everyone, thank you very much for the kind introduction. I want to welcome everybody to today's webinar. And today I want to talk about the battle between plants and viruses, or the counter battle between viruses and plants, and especially about transcriptomic analysis and how we use microdissected plant cells to analyze turnip mosaic virus-infected cells. And just let me briefly introduce myself. So my name is Björn Krenz. I work at the DSMZ. That's the German collection of microorganisms and cell cultures. As you can see in the upper left part of the slide, we are located in the northern part of Germany, and underneath you can see our building. So my office is right here at the left lower corner. And as I said, we are a collection of bioresources. You can deposit, order, or we provide bacteria, for example, human cell lines, phages, plasmids, and also plant viruses. And that's actually where I'm affiliated with, where I'm associated with, is the plant virus department. So we take care about plant health. And besides that, we have a virus collection, which you can use for example, as a positive control in your ELISA tests or serological tests. Yeah. We offer diagnostics, including also high throughput screening. So whenever you have a sample which is, and you suspect it's a virus infection, you don't know what it is, send it in and we can tell you. And just let me briefly introduce my group. So we are not focused on the service, we are focused on research. And my research group consists of several people. So we have Verena Mayberg, for example, as my technical assistant. We have Enneke, which works on the CRISPR-Cas project, and then we have here Maria and Hendrik, both PhD students. Maria is working on nanoviruses. Hendrik is working on a cell biological topic called stress granules and the role of stress granules in virus infection. And then we have Khalid, and Verena, they did most of the work at the laser dissection microscope. And Khalid, which is not very surprising, he has now his own group. Yeah, but we have an additional PhD student here, Christoph, and Christoph is working on Gemini viruses, a very important plant virus family. So why do we study plant viruses? Yeah, simply because they can cause severe diseases. So, for example, this tomato disease, which happened like 2 years ago, which was very severe for the tomato growers here in Germany and besides Germany. Or here you have an example from the field. It's a pea field, and you can see this yellow spot here. So that's an incident of the nanovirus infection, which actually destroys the pea harvest. Or another example, which is not restricted to Germany or to Europe. In former times, I worked, for example, for the on the red blotch disease, a virus which causes significant crop loss in grapes and for the wine production in the United States and beyond. So how do we study plant viruses? Yeah, and so today's webinar is about the laser micro dissection combined with a smart sequencing technology to conduct the early transcriptomic analysis of Arabidopsis thaliana infected with Turnip mosaic virus. So today's webinar is about the early events of infection in plants. So this is why we use the CellCut or selector system or the laser dissection, because we want the very first cells. We want to analyze the very first cells because this is the onset of the infection. Yeah, and this is what we need to analyze, because when we can prevent the onset of infection, then we might have a chance that the whole plant does not get infected systemically. Yeah. And one analysis we are doing is, for example, a comparative transcriptome analysis. So this is a picture of the system that we use here in Braunschweig. Yeah, you can see on the left part. So we have a combined system of a confocal laser scanning microscope, and we have the CellCut and the selector MMI system. Yeah, so 3 systems in one. We can make nice pictures, but we can also cut out tissue, or we can use the selector system. And I will introduce both systems, but with the focus more on the CellCut. Yeah. So why do we need the CellCut? So why do we need to cut out cells? Yeah. So when we look in an infected leaf, and here this is a sketch of a leaf, you can see there are several cell layers. Yeah. You have for example, the epidermis, you have the mesophyll cells, you have the lower epidermis, you have veins. So a lot of viruses, they're only located in the veins. So we really need to go to these cells. We need to dissect these cells and analyze those cells. And this is what we use. And as I said, for example, you have something like stomata, for example, at the lower epidermis. And this is now an example how we cut out stomata. Yeah. So what you see here is a microscope picture of a peeled epidermis, lower epidermis of a tobacco plant, and you can see this mouse-shaped cells here. So these are 2 cells. This is the stomata. Yeah, and besides you have the epidermis cells, and you can see this puzzle-like structure. And now we only want to cut out this stomata. Yeah, so this is a proof of principle. Yeah. Just to show you an example how we gain those cells, what we really need. Yeah, you can see here the laser is working. It goes around the stomata, and then the tubes come down, and we collect the stomata. Yeah, and this is now in our collection tube, and you can see here that's the rest of the epidermis. Yeah. And as I said, in former times, Khalid did most of the work. Nowadays, it's Verena or me. And so Khalid was especially interested in the veins of a plant or in the phloem. Yeah, and which is, as when you recall the sketch I showed you before, it's within the plant. Yeah, so we need to embed the tissue. We need to make sections. Yeah, but we also need a marker to show the phloem, yeah, to visualize the phloem. And Khalid used the fluorescence marker. Here it's GFP. Yeah, and what you can see here now is a section, and this is the phloem glowing. We have a special marker, GFP expression marker, green fluorescent protein in phloem. And so he marks now the area and cuts out, and the GFP is bleached because of the heat of the laser. Yeah, but you can nicely see that the laser goes around the phloem, and then afterwards, when the laser has done its work, we collect this tissue in the tube. Yeah, and then we can further analyze the tissue, and what we have now is an enriched sample of phloem tissue, and we can compare it to other tissues. Yeah. So the thing is, we want to investigate the plant virus or infected tissue. Now we combine the thing. Yeah. So what we do now is that we alter or attenuate the virus in such a way that expresses, for example, GFP, like in this picture here. Yeah. So what you have here is a section of a petiole. Yeah, you can see here the hairs. This is the epidermis. Yeah, and already after 1 or 2 days post-transmission, you can see the infection site here at the epidermis. So, I mean, this virus was altered that it expresses GFP from its genome. So every infected cell is now glowing in green. I show this here in red because of the contrast. But this enables us to really identify infected cells at the very onset of infection, already 1 day after transmission or 1 day after infection. Yeah, and at the lower panel you can see the situation after 4 days, for example. So this is the whole leaf under UV, because the chlorophyll is red under UV. Most of the leaf is red. Infected cells are in green. You can see the green vein. So this is the tip of my thumb on the lower left part. And 2 days later, you can really see the spread of the virus. Yeah. So this is already in a systemic infection. Yeah, the plant gets fully infected. Yeah. But what we want here are the very first cells. This is what we want to analyze. So the tool we use here is that we have a virus, for an example, which expresses either GFP or here for the Turnip mosaic virus, you will see it's RFP, which enables us to identify infected cells. Yeah. And the virus I want to introduce now is the Turnip mosaic virus, which infects Arabidopsis thaliana. So Arabidopsis thaliana is a model plant. It's well investigated. It's a very easy genome. On the right panel, you can see the virus as an electronic microscope picture. It's quite long. It's filamentous. Yeah, and here you can see a close-up what it can cause to Arabidopsis thaliana. So here on the left side you have a healthy plant, and the right side, yeah, you can see the yellowing, or yeah, how the leaves are deformed caused by this virus. Yeah, and below the picture you can see the sketch of the genome organization of the Turnip mosaic virus. So this is a single-stranded DNA virus, a plus-stranded RNA virus. So this RNA, this viral RNA, can be immediately translated into a protein, and it has a polyprotein strategy, which is then cleaved in several other proteins. Yeah. And now imagine we sneaked into this genome the open reading frame for RFP. So when we now infect the plant, then we get red spots under UV. Yeah, so we can really see here. So this is an infection site, 2 days post-infection. Yeah, and this is 100 micrometer. You can see the scale here, and when we zoom in, we can see here the epidermis cells, this puzzle-like structure, and this bright red dots in between. That's the nuclei. Yeah. And this is exactly the tissue what we dissected. Yeah. And so the pipeline here is, well, it's not simple, but just to show you the pipeline or the workflow, well, in total we had 5 replicates. We had healthy controls. That's for clear. We did this several times. We prepared the libraries, yeah, and sequenced the libraries with the smart sequencing protocol. Of course, we need to process the data, all the bioinformatic pipeline, and ended up with the functional classification of differently expressed genes. And what you can see here, that's the mapping statistics, how many reads we had. We have a sequencing, we have the sequencing facility in-house. So if you want to do high throughput sequencing, you can do it here if you like. And in total, yeah, to give you some results, we have several differently expressed genes. We have a lot of genes which have been upregulated. We also have genes which are downregulated. We have 321 total upregulated and 193 which are downregulated. To a certain extent, yeah, you can see the conditions here on the slide. And then when we have a closer look, and usually when we do a GO ontology analysis, yeah, you have molecular function and kind of stuff. And here I only want to show you the biological processes. And so this is not really unexpected, but it's really nice to see that, for example, in upregulated genes, you have something like immune system process, regulation of response to stress, response to oxidative stress, response to fungi, to viruses. Yeah. So and cell death. So this is actually what we would really expect. Yeah, that we see a response to viruses or to a pathogen. And on the other hand, we have a downregulation. You can see here, for example, photosynthesis is downregulated, which totally makes sense. Yeah, and other stuff. But now we really need to validate if this is really true. Yeah, if we can rely on our RNA-seq data, and this is what and this was proved by an additional validation by QRT-PCR. So after 2 and 7 and 14 days, you can see an increase of the virus. Yeah, it goes up, which also makes sense because the viral titer should increase. And then here on the right panel, you can see that we have different genes. Yeah, I erased the genes because we want to keep this confidential so far. But if you have further questions afterwards, I can give you maybe more details here. But the thing is, we validated, and you can see that the genes which are really upregulated over the time are upregulated or downregulated over the time. So this really proves our RNA-seq data. We can rely on our RNA-seq data, and we can also see that this kind of expression, 2, 7, 14 days, really changes over time. And this is quite important, or this really shows us why it's important to see a very early event of infection. Yeah, because when you wait 5 days, 7 days, 14 days, the story is completely different. Yeah, so here we can really assess the response of the cell immediately after infection. Okay, so this was the results so far. What we have for the Turnip mosaic viruses analysis, we have some more. For example, what we have more is, or what we can do is here at the DSMZ, when we use the laser dissection microscope, the CellCut system, that, for example, that we play a little bit Jedi Knights, for example, when we divide oscillatoria. And so a colleague of ours, they approached us and said, well, we have this oscillatoria here, and we need to propagate it, but it does not work. And propagation happens when you, for example, just split one into two. And they asked us if they can use the laser just to split them into two parts. And this is actually what we did. So this is an oscillatoria here, and we used the laser just to cut it and to make 2 out of 1. And yeah, this wasn't really complete, so another cut, and it really worked out. So after several days, we could actually see that those parts were growing, and that we can use the laser or the CellCut system to actually propagate the oscillatoria, in a very quick way. And then, in addition to the CellCut system, we have the selector system, which is a capillary-based system. And I just want to briefly show you how we use the selector system here. And as we work with plant and plant tissue, we also work with protoplasm, which is actually plant cells without a cell wall. Yeah, and you can see in this picture a protoplast suspension. Yeah, so some protoplasts, they are bright green because there are a lot of chloroplasts. Some are nearly transparent because there are cells that don't have much chlorophyll. And we want to collect just one of the cells, just one, because because you just need one protoplast and you put it in the proper media, you can generate a whole plant out of it. Yeah. When you think of CRISPR-Cas and genome editing, yeah, and then you can just pick the right protoplast you want, yeah, with the correct editing and propagate it. And so here we use the select a system. Here's heat capillary. Yeah, we approach the protoplast, suck it in, and then we want to deposit the protoplast in a new vial. And here we press it out, and it's in the media. Yeah. So this is basically what I wanted to show you in today's webinar. I assume I was quite quick, but anyway, I want to thank you for your attention, and I'm happy to take any questions afterwards if you like. And thank you for joining the webinar. Thank you.
Well, thank you very much, Björn.
You're very welcome.
We've got a few questions from the audience.
Yeah.
So let me start. We've got quite a lot of questions about the laser cutting, and then a few questions about the upregulation, downregulation results that you showed. Mm-hmm. So let me start by asking you, were you using any RNase inhibitors to prevent RNA degradation during laser dissection?
So actually, this is a little bit a tough question, because what we use for RNA extraction is a kit. It's called a GeneElute FFPE RNA purification kit, and it has some RNA inhibitors, yeah, but I can't tell you exactly which one.
But they're there.
They're there, yeah.
And does the cutting damage the guard cells and induce a wounding response?
When you use fixed tissue, there's of course no wounding response because everything's fixed. Yeah. Mm-hmm. So in this example where we were cutting out the stomata, it was peeled epidermis. It was live, or was the tissue was still alive or non-fixed. And here you have to place the laser not very close to those cells, yeah, because the heat of the laser, of course, will cause a reaction and will damage the cells. Yeah, and so you must keep in mind when we cut out tissue or when we cut out cells, it's an enriched fraction. Yeah, it's the cleanest what you can get. But in scope of your experiments, you always have to keep in mind that the cutting and the preparation has an influence on the outcome, on your results. Yeah, but you can't, until now, the technique, that's what we have, and it's the best what we have. Yeah, so it's the best result we get. Yeah.
Yeah. So
okay.
I think you also answered someone else's question in the meantime, okay. Because we got a question asking if you used fixed samples, but I guess you're saying you can use fixed samples, but you didn't here.
Yeah. So for this experiment with the Turnip mosaic virus, yeah, so we basically used FFPE fixed tissue, so formalin fixed tissue. Yeah. But for example, when we cut out nuclei and we used a peeled fraction of the epidermis, then there's no fixing. So we basically use both.
And when you're doing the laser cutting, is the cutting
mm-hmm.
path set beforehand and then automatically done?
Oh yeah, so this is pretty cool. So basically, this is an automatic system. So what you can do is you can make a picture of your whole slide, yeah, and then you can begin just to mark the areas which you want to cut out. And you can say this is area 1, this is area 2, this is area 3, and you can cut out all at once or subsequently, for example, and you can place sample number 1 in tube number 1, and you can place a sample number 2 in tube number 2. So you have a lot of options, yeah. And you can either use your mouse, your pointer, and draw, or you can just say, okay, I want to draw a circle or a square, or you can use a pen, yeah, and can actually draw on your screen the area you want to cut out, and then say, laser go.
Mm-hmm. That's very clever.
Yeah.
You also showed some results of upregulation and downregulation of different genes. We had a few questions about that.
Yeah.
One was, what are the various mechanisms that you use to conclude which ones are upregulated and which ones are downregulated?
Oh, so basically this was a comparative analysis. So we compared infected to non-infected tissue. Mm-hmm. Yeah. And everything was prepared the same. So we had a mock infected plant. So mock means that we more or less treated the plant the same way as with the virus, but without the virus, and then cut it out in an area similar to the size and leaf stage to the infected one. Yeah, and then we compared those two samples.
Let's see. And we also had a question about, I don't know if you know this off the top of your head, but what was the p-value for that experiment?
Oh, the usual p-value, I think it was also in one of the slides. That's 0.05.
Okay, great. Sorry, I'm just scrolling through all the questions. We've got quite a lot that came in the last few minutes.
Okay.
We had a few more questions about the laser cutting. What is the smallest fragment you can cut? So what is the resolution of it?
Oh, wow. Okay, so the resolution of the laser, if I got it right, yeah, and hopefully MMI won't sue me now. No, I'm kidding. So when you use, for example, 100 times of objective, we were able to cut out something which was like in diameter, roughly a micrometer. So when I first tested the system, we were cutting out chloroplasts from peeled epidermis cells. And chloroplasts are usually in epidermis, they are roughly, they're like lentil shaped, but they're roughly like 5 micrometers, and it worked nicely. So, and then we were cutting out bacteria, and they had the size of a 2 micrometer. Yeah. And we were able to not only to isolate the bacteria, but they were also still alive, so they could grow. We pushed it to the limit. So plant cells, they have cell-to-cell connections. They have plasmodesmata. They have cell-to-cell connections. They're roughly like 0.51 micrometer. And we were able to cut it out.
Wow.
But you need a very good objective for this.
Yeah. Yeah. And how were you able to validate that cutting does not have any influence in the expression?
Well, the validation is always when you use the control. Yeah. And so when we cut out tissue, the laser also was cutting out the tissue. So the influence of the laser and everything, the whole procedure, yeah, also happened to the control. I mean, as I said, so far, in my opinion, that's the best technique we have so far to gain from a single cell, for example, an organelle like the nucleus. Yeah. Yeah. And of course, the preparation and the sampling, it has an influence. But with the proper control, I think you can make a statement. Yeah. But that's up to the reviewers. That's my opinion.
I've got a few more questions that came in after the talk finished, so yeah. I'll ask you a few of those. You mentioned micropropagation at one point, and someone asks what that is. Can you explain that?
Micropropagation. I'm not quite sure if this refers to the oscillatoria. I hope it refers to the oscillatoria. So for example, or it was with the protoplast. That might be correct. So, for example, when we want to genome edit a plant, yeah, there's one approach that we prepare protoplasts, for example, tomato protoplasts or potato protoplasts, and then we transfect the CRISPR-Cas system into those protoplasts. And when you highlight now the Cas9 protein, which is the key enzyme for genome editing, for example, with a fluorophore or a marker, whatever, and you can actually identify which of the protoplasts have been transformed, then we can use the selector system to specifically pick those, deposit it into a new vial, and under perfect conditions, which means the proper media and then phytohormones, that kind of stuff, those protoplasts, they can actually start to divide, and they're forming colors. And then again, with the perfect or with the optimal phytohormones, out of the colors, you can generate a new shoot and you can generate a new plant. Yeah. Mm-hmm. Or you select bacteria. You have, for example, yeah, you have a sample from the environment. You have different kind of bacteria. You want to know which bacteria are in it. Yeah, and you just use the system either to cut out from the slide, or use the selector system and pick a single bacteria, and you propagate them in a reaction tube and grow them, and then you can analyze them. I hope this answers this question.
I hope so too. So I'm just going through the list of the other questions and comments that came in. I'll pose you a few more questions. But in between the messages, I see there's a message from Khalid, who you mentioned in your talk, who did the experiments, and he's been listening to the talk. He says he's happy to help answer questions. So what I want to suggest, there are a lot of questions that we won't be able to get to in this talk, and we will forward all the questions to you. So if there's anything, then Khalid can help answer those as well. That might be a good idea, because some of them get very technical as well. Okay, let me go back through the list, because there were a couple of questions from different people who want to know if you can use this in animal cells or in human cells as well as in plant cells.
Of course. Sure. Yeah.
I guess everyone is asking about their own cells.
It's actually much easier, I guess, when you have cell cultures. Yeah. It's actually a little bit more difficult for plant tissue, because plants, they have the cell wall, and the cell wall is quite thick, and quite strong, so sometimes you have to drill with the laser, to get your sample, so with the cell culture, it's a little bit more easier.
And I noticed a few people were also asking about the depth of how far the laser can cut. So someone asked, could you make a whole cross section of a cell using this technique, or does it only go to a certain level?
So you mean the how deep the laser goes? So how thick the okay, as I said, so what we use is when we embed tissue and we make cuttings with microtome, we usually have like a thickness of 10 micrometer. Okay. And so this is quite easy for the laser. For the peeled epidermis, it's a little bit thicker. But in any case, so what the system can is, if as I said, there's a drill function, so you can adjust this, that the laser cuts the area, and then you alter a little bit the focus of the laser, and you lower the focus, and then you can cut deeper. So by this you can actually drill into tissue with it, which is quite thick. And or you can just increase the power of your laser, for example. That's also one possibility. So when you want to collect your sample in the beginning, you have to make sure you have the perfect conditions. Of course, there will be a limit. A sample can't be too thick. But if you're in the range, I think like from, I don't know, to 20, 30, 40 micrometers, I think you're fine.
Okay. Let's see. There's a question about the RT-qPCR. Which normalization gene was used, and how was the selection done for a relative gene expression study?
Oh, see, now I need Khalid. So what I can tell you. So what we use is TuMV-infected tissue. Okay, and the virus. So usually you use a housekeeping gene. Yeah, you use something like, for example, actin or something. But that's a different story when you work with viruses. Yeah, so a virus can alter expression levels of housekeeping genes, or what you usually use as a reference. So there are publications. You can look up which reference gene is perfect for your virus. Yeah, and I'm not quite sure if you use maybe also a worky gene or something like that. But so that's the question. Yeah, Khalid needs to answer. or I must look it up.
Yeah, Khalid, you can leave your answer in the questions box, and I'll pick it up from there if you want. I'm just going through. We've got a lot of instead of comments, people have left a lot of instead of questions, they've left comments to say thank you, and we even had a message from someone who graduated botany in 1973 and is fascinated by the technology.
Oh, cool.
So here's another question for you. You used GFP expressing virus to infect plant cells isolated by MMI. How is this technique advantageous over cell sorting by FACS?
So cell sorting means that we need to prepare protoplasts. And to prepare protoplasts, we have to remove the cell wall. So actually we have to disrupt the tissue. And that's, of course, a disadvantage. I totally understand FACS people. I did it also in former times. So for former times, for example, we were actually collecting fluorescent nuclei. So we prepared the nuclei and then we enriched the fragment by FACS. But prior to your analysis, you have the preparation of the nuclei. And so when you think of a plant cell and a virus-infected cell, then you really want to investigate the virus in its proper environment. So you actually don't want to really disrupt the tissue, especially, for example, when you want to investigate cell-to-cell movement, for example. Yeah, for example, the plasmodesmata. And so here I really see an advantage when I fix tissue, cut it, and use the CellCut to get my sample.
I see.
I hope that helps.
I'm sure it does. We had a few questions asking if you're using fixed cells or fixed samples, how do you prepare them before cutting?
Here you have to look up the protocols. I think this would be a perfect question to answer afterwards. Also with for Khalid. So usually we use formalin fixed tissue when we want to go for the RNA. I think it depends what you want. Yeah. So for example, if you want a proteomic analysis, yeah, you need a different procedure to preserve your tissue. And when you go for the RNA, you really want to make sure that the messenger RNA is not degraded and that kind of stuff. If you go for DNA, for example, it's much easier. So it really depends on the application or the analysis you want to do afterwards. Yeah, and I think in the meantime, when you look up, there are several protocols and several very good protocols which give you an idea what kind of fixation or preparation prior to cutting you should use.
I see. We've got a question about the fluorescence tag that you used in the viral clone. How stable is this fluorophore?
So what we use is, for example, for the TuMV. So this was mRFP, so monomeric RFP of and this is quite stable. So the lifetime of GFP or RFP is quite high. Of course, you can use other labels, but I must say we had good experience with GFP or RFP.
And someone asked, how do you choose the location where you were cutting? How do you make that decision?
See, that's the reason why we use GFP, not RFP. Yeah. So under the UV light, so at daylight, the leaf is bright green. Yeah. But under UV light, the leaf is bright red because of the autofluorescence of the chloroplast. But when you use the proper filters and the proper excitation with your microscope, with your fluorescence microscope, and that's the reason why we have a combined system of fluorescent microscope and CellCut. Then you can see the GFP highlighted cells or the RFP highlighted cells under UV. There are other possibilities. You can make sort of a staining, for example. But when it comes to plant viruses, we had best experience with GFP and RFP or other fluorescence proteins.
Thank you. We've got a few questions left to go. Let me have a look because I'm going through the list in a different order. So these will be your last two questions. What is approximately the lifetime of GFP and RFP in these experiments?
Oh, that's really tough to estimate. But I think I can recall a study where they say the turnover of GFP is between 24 and 36 hours. So when you stop expression, even 1 or 2 days after you stop expression, you will see GFP. But
yeah,
yeah,
that's long enough.
That's a question for Google. That's a question for Google.
But you haven't noticed anything fading during your experiments, so it's.
No, I mean when you use a free GFP, for example, yeah, GFP is it's 27 kilodalton, and it can move from cell to cell, especially in the plant virus background, because viruses usually they increase the size exclusion limit of the plasmodesmatous. Proteins can flow to the adjacent cells. So if, for example, you use a GFP or RFP with a nuclear localization site, then it's or with the ER retention signal, then it's stuck to the ER or it's stuck to the nucleus, and then you have no fading. Yeah, and but of course with the heat of the laser, yeah, it fades away. Yeah, you have some bleaching.
And then your final question is: you mentioned that you isolated the nuclei of the plant cells.
Yeah.
Did you laser cut these, or did you cut whole cells and then separated the cytoplasm from the nuclei?
Oh, we directly isolated the nuclei. Yeah. So it's I can't really show a picture here, but so when you have the epidermis cell, and then you can nicely see the nucleus. The nucleus of an epidermis cell in tobacco is roughly 10 micrometers. So it's not that small. Yeah, and you can nicely draw a line around the nucleus, and then the laser cuts, and you can collect the nucleus right from the sample. And actually, what you can do is you can compare, for example, a cytosolic fraction compare with the nucleus fraction from one cell, if you like.
I see. Well, thank you very much. I know there were a few more questions, and some of them were quite specific, so I will leave those for you to look at a later time. And thank you again for a very illuminating presentation and a great discussion. And thanks also to the sponsor, Molecular Machines and Industries. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you have enjoyed this seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com, and it should be available within the next 24 hours. And when you're there, you can also see other webinars we have lined up for you. So until next time, good luck in your research, and goodbye from all of us at Molecular Machines and Industries and Bite Size Bio.
Bye.